Hello and good afternoon. This is Fred Heinzel. I'm an associate professor of USF College of Medicine, and I'm going to talk about the microbial pathogenesis update. It's a bit of a grab bag of topics, but let's start. Okay, first off, there's this long story about vitamin D and tuberculosis. And in the last several months, there have been several articles in Science, Journal of Bacteriology, and other locations that have beginning to point a finger at vitamin D as being important in anti-tubercular immunity. Now, observations of vitamin D and tuberculosis go back quite a long ways. And these weren't exactly controlled studies, but there was a Dr. Hermann Bremer who was a German biology student who unfortunately had TB. He thought that he didn't have long to live, so he thought the most exciting thing he could do was go to spend his last days in the Himalayas. Unexpectedly, he did better, and he attributed this to the fact that when he was in the Himalayas, it was wonderful, clear weather, high altitude, that he had received a lot of UV light, and he thought somehow light was helping his TB. When you connect this to another observation by Dr. Finson, who won the 1903 Nobel Prize, showing that you could cure cutaneous TB with UV light, some other types of lights went on, and this led to the idea that that vitamin D generated by light what might in fact be important in resistance against TB. This still remained anecdotal. The sanitarium system, which actually was set up by Dr. Bremer when he actually had come back, emphasized fresh air but also sunshine therapy. Of interest, at this point they knew that rickets could be treated with cod liver oil or other vitamin D containing compounds, and that they benefited when they were in the sun as well. As it happens in these sanatoriums, you would have one group of patients with rickets and another with TB, and both actually seemed to improve on the same regimen of either vitamin D or lots of sunshine. And eventually, it led to the use of single or multiple dose vitamin D, which similarly improved both rickets and TB. Unfortunately, none of these were really studied in a way that allowed any sort of measures of significance. Since then, moving forward to 1970 to 1980, there was a case control study showing that vitamin D deficiency in a specific population of Gujarati Asians who, because of dietary prescriptions, took in very little vitamin D. They were known to be vitamin D deficient. They had a lot of rickets in their population. But then there was a case control study showing that they in fact had a markedly increased risk of active TB compared to controls. The odds ratio was as high as 2.9 and was significant. So this led some of these more recent studies to address the role of vitamin D in resistance to tuberculosis. Now, there are a couple of other methodologies that have been tried. One is looking for putative mycobacterial susceptibility genes. One of the methodologies is a genome-wide scan, where you essentially use primer pairs to identify polymorphisms that are spread across the entire genome. More recently, you can use single nucleotide polymorphisms for that, sometimes using chip devices, which allow you to obtain a lot of data in a more expeditious fashion. But from a couple of different studies, and this one I think was from Oxford, it was shown that tuberculosis, in terms of susceptibility regions, it was highly associated with the vitamin D receptor, with genotypes that affected the promoter function, either a genotype FF or capital TT, capital T, little t, which resulted in changes in the promoter function. And suggesting then, providing additional evidence that vitamin D, and specifically the vitamin D receptor, was involved in anti-tuberculous immunity. Okay, since then there's been bench research really nailing down some of the mechanisms of this. In this study, Dr. Martineau actually took 196 PBD positive patients, randomized them into placebo and single-dose vitamin D groups, and essentially studied killing of a type of Mycobacteria in a whole blood culture system. The hypothesis being that vitamin D would increase mycobacterial killing in vitro. Recognize that the previous studies just showed there was an anti-TB effect. Here they're hypothesizing specifically that it is an immune-mediated killing. Okay. Please note that the amount of vitamin D these patients take was very high. It was almost 100,000 units, and not something I'd recommend to do on a regular basis. After 6 weeks, which gave the T cells a chance to maybe incorporate whatever vitamin D does, they went into an in vitro assay to study killing of Mycobacteria. Now, they didn't want to work with viable Mycobacteria TB, and they wanted some mechanism where they could have a very rapid readout of the number of bacteria killed. So they used an assay which is using BCG, Mycobacteria, which is fortunately not particularly virulent on the benchtop, and it's a locus assay. What it is, they engineered in a luciferase gene into BCG, and in the presence of luciferin in the culture media, these will glow intensely, as shown on the right-hand side. And in order for those to glow, the bacteria has to be viable. It has to be able to make ATP to actually make the luciferin glow. And so that tells you they're viable. So this was a great assay, and when they tested the vitamin D treated patients, the luciferase activity, in other words, the viability of the BCG, was diminished by half, which was highly significant in the system that they had. It had no effect on interferon gamma production, which they measured as well. Both the control and the vitamin D group made significant amounts of interferon gamma to the BCG challenge. Okay, so how does the vitamin D increase mycobacterial killing? Big question. On the left-hand side, it's fairly certain that it's going to be T-cell mediated. So this additional group now focused on the role of T-cells and vitamin D receptor. On the left-hand side, you can see the two cells. The one on the bottom with the red in its periphery is a T-cell, and that's basically just standing for a T-cell receptor. And then there's a target cell on top. On the right-hand side, we're looking at the basic mechanism of T-cell receptor activation. On the top, in that bilayer membrane, you have a T-cell receptor. When it interacts with a TB antigen, it sends a signal that results in production of the vitamin D receptor. The vitamin D receptor is, of course, in the cytosol. When it encounters vitamin D, which was, in this case, provided dietarily, the occupied receptor now translocates into the nucleus, and from there it acts as a transcription factor, and it induced production of an important regulatory enzyme called phospholipase C gamma 1. This has an important role in a cascading series of protein kinases, particularly what they call the MAP kinases, which are very involved in T cell and any other cell function. From the result of that, the ability is fed back, and the T cell receptor, when it encountered TB antigen this next time, had as much as a 100-fold increase in receptor activity. So in this study, it looks as if the vitamin D receptor bound to vitamin D, in fact, it's like a rheostat, turning up the gain on the T cell receptor, which of course would probably improve whatever function the T cell was providing. And another group addressed more specifically how is this killing accomplished. The T cells and macrophages, to some extent, have a variety of lysosomal structures that contain enzymes, peptides, and other particles. These include an antimicrobial peptide called Cathelicidin, which is upregulated in the setting of a highly activated T cell. It is a peptide that then, when it's released, is capable of killing a variety of bacteria, and confirmed by others is that, in fact, Cathelicidin does in fact kill M. tuberculosis in cell culture. and I think there's a piece missing there. To confirm that it was actually the Cathelicidin that was killing the Mycobacteria, they used what they call siRNA, which is a way of essentially targeting all of the RNA making Cathelicidin for destruction inside the cell. In other words, it no longer makes Cathelicidin, and when they studied that particular treated T cell population, they were unable to kill the tuberculosis, suggesting that in fact the Cathelicidin is sufficient to kill TB. There's some other interesting aspects of Cathelicidin. One is that it had been associated previously with protection against infection in renal dialysis patients. And I'm not sure why they have high levels, but they had several groups, and they identified that the amount of Cathelicidin in sera apparently was protective, although probably was actually inside the T cells and macrophages. Okay, so that's news. So vitamin D helps us kill Mycobacterium tuberculosis. I don't know, we should all go and immediately start ingesting 2.5 milligrams of vitamin D, but knowing that mechanism, it may be possible to perhaps somehow develop a Cathelicidin-based treatment of disease. But I'm going to move on to another topic now, which is what is the role of the tuberculous granuloma in an infected individual? It's been held for years that it's a way of encapsulating the bacteria so they can't cause trouble, and that this is a good thing for the host. There's been some suggesting recently maybe it's some other way around. So on the left-hand side, you've got two different examples of a fairly early granuloma on the top, one on the bottom that clearly has caseous necrosis. And on the right, you see a zoomed-in image of that epithelial layer, which is actually, these consist almost entirely of macrophages that are kind of stretched out and epithelial-looking. So the story of the granuloma is tied in with, I think, a very important virulence mechanism by which Mycobacteria works. And this is encapsulated into a single area of the Mycobacteria genome called the RD1 virulence locus. And I think I've got this all set up. So down at the bottom, under B, here's a picture of the gene. It's just a piece of the BCG chromosome, by the way. But genes that of interest include these two here. These constitute an ATPase, so an energy, basically a little motor that associates with a secretion pore in the membrane of the TB organism. Now, what's secreted through there are these two proteins. One is CFP10 and the other is ESAT6. I can't tell exactly what they stand for right now, but ESAT6 in particular has been suspected to be an important pathogenesis factor for some time. And those are transported out of the secretion apparatus and actually then are able to get into the surrounding medium outside the cell as well. So what's interesting about this is the presence of an RD1 locus seems to indicate pathogenicity. For M. bovis, which was a precursor strain of Mycobacteria from whence BCG was generated, M. bovis was actually a reasonably virulent organism and it caused intestinal TB. You can rarely see it in some areas of the world still, but it was a very serious pathogen. At some point in its passage in some lab somewhere, that RD1 virulence section got taken out, and that strain then was realized. They realized it was an avirulent form of M. bovis. It was called BCG from where it was derived. So RD1 again encodes production of T cell antigens, and it's not just that they're spitting out proteins. These proteins are actually already recognized on the basis of twin studies to be important in the generation of symptomatic TB. And the secreted protein ESAT has been suspected as being important in immunologic TB response, as had the cultured filtrate protein 10. In fact, fact, those are the two antigens that are tested for in the quantiferin gold assay. What's even more interesting is that ESAT6 production has been shown recently to regulate granuloma formation. So it's not just simply a protein that gets secreted out. That's a very interesting antigen that our T cells see, but rather it actually seems to have distinct effects on tissue organization. So it begs the question, are TB granulomas, for instance, if the Mycobacteria in fact is actually trying to orchestrate a granuloma, is it possible that TB granulomas are protective to the pathogen as sort of a Trojan horse mechanism, or is it protective to the host? So to get to this question, it was a little hard to use BCG, it was a little hard to use MTB. Those models would involve mammals and were very hard to work with. Instead they went for another model, which is the Zebrafish model. Now recognize that Mycobacterium marinum is a Mycobacteria has an RD1 virulence locus. Yes, it causes disease in humans occasionally. What it is it's fish tremble in fear at the thought of Mycobacterium marinum because they develop a form of fishy tuberculosis. And in fact, it's they get vertebral abscesses and they form this hunched back. And there's actually a name for that disease. I just can't recall it right now, but that's a big issue with fish. So Mycobacterium marinum, zebrafish, is like TB to humans. You get classic granulomas. And here's some surprising facts about fish, is they actually have T cells. And you have an alpha beta T cell, you have gamma delta T cells. You can actually induce a REG1 mutation. REG it's a recombinase activating gene, and what it does basically is you take all those little pieces of the T cell receptor and they all have to be spliced together so you have the intact recombined gene. If you take that out, they cannot make those T cell receptors. You can't make B cell receptors with it. So the REG1 mutation conferred extreme susceptibility of fish to Mycobacterium marinum, which is what you would it's in parallel to what happens in humans who don't have effective T cells to fight tuberculosis. So now you have a fish model of fish tuberculosis that is probably suitable for answering some very basic questions about the disease. It's a very interesting model, by the way, for other things. It really took off because it's a fantastic system in which to study morphogenesis during, from embryo forward. And the embryos are transparent. And if you want, you can engineer in green fluorescent protein behind a gene in these fish. And every time that gene turns on, you see a big green spot. And so for neuroanatomy, you can basically assay which neurons are being secreting which protein at a given time in morphogenesis. So it's a very cool model. At Research Day over at USF, there were no fewer than, I think, 4 posters on the zebrafish model for Bartonellosis was one of them. Okay. So here are the granulomas of Mycobacterium marinum in the zebrafish. You need only 5 colony-forming units, and these fish generated a fair number of granuloma. And on the bottom, you can see these are pretty typical. The one in the middle with the black arrow and the other one next to it both show caseation necrosis inside the granuloma. You got sort of an epithelioid layer around that one. And going up in the top, which is an acid-fast stain of the tissue, you can in fact make out numerous acid-fast bacilli. And in the granuloma, you can see actually granuloma is full of acid-fast bacilli in that picture, right? The picture's up on top. So this is a great little model to work with. You get classic granulomas, you can actually see the acid-fast bacteria. Okay, so they make granulomas. The question came up is what happens in these infected fish if they're infected with a non-virulent Mycobacterium marinum strain. In order to develop one, they went and deleted RD1 locus from M. marinum. So essentially they made a fish BCG equivalent and tried it out in the zebrafish host. Because you took out the RD1 locus, you don't make ESAT, you don't make the CFAP10, which actually is fairly focused in terms of knocking out some proteins. There's really only maybe 2 proteins or 3 proteins being made by that area. So if you have a change in phenotype, then the next job is going to be easier. So what happened in this? Macrophages infected with virulent Mycobacteria marinum release ESAT6. ESAT6 was found out then in this model that it stimulates epithelial cells around the granuloma to produce matrix metalloproteinase 9, which is just a proteinase that eats up glycolated proteins that are important in gluing cells together or providing interstitial structure in tissues. But what they found is that when the matrix metalloproteinase chewed up the matrix, this generated small compounds that attracted new macrophages that in turn became infected with the mycobacteria when they arrived there. Curiously, the granuloma expanded and the bacteria proliferated. This is not what they expected if you thought that the idea of the granuloma is to protect the host. It looked as if actually this was an environment in which the bacteria were proliferating and doing very well. So what they did is they then took the RD1 deleted mutants of M. marinum, which fail to make ESAT6 and don't make granulomas. And what they found is that the mutant M. marinum numbers actually decrease, suggesting that ESAT6 was necessary for bacterial proliferation, which suggests then that the tuberculous granuloma may in fact be a place where significant proliferation of bacteria takes place, and it's truly a Trojan horse system. Okay, so the granuloma is not a host defense response for containing an infection. Instead, granuloma formation benefits the pathogen rather than the host. Recognizing this is all derived from this one zebrafish M. marinum model. However, the proteins involved in the pathogenesis are relatively few and are highly conserved between those two bacteria, so it's likely there's some validity to this. There's some things you can, I think novel ideas can come out of this. One is, of course, we're all worried about hyper-resistant MTB, extremely resistant MTB. And we have very few new drugs with which to challenge hyper-resistant TB. Instead, this suggests that maybe another way to go about this is really to work on some of these factors. One possibility is designing a new type of vaccine based on ESAT6 and CFP10. This makes sense, and it's been proposed actually for several years because, again, these are highly immunodominant proteins recognized by T cells, and they actually do seem to mediate some of the pathology of disease. So is it possible we could just generate neutralizing anti-ESAT6 antibodies, or do we vaccinate against those particular proteins, and so that we have a stronger T cell response to those targets? Knowing that matrix metalloproteinase is in fact involved in this process is of interest. Turns out that if you want to inhibit metalloproteinase 9, you can actually, doxycycline or any of the tetracyclines have some inhibitory properties. There are also some, I think in the pharma system, there are some specific inhibitors of all the metalloproteinases that are coming out. And it's likely that we might find some unusual and novel approaches to inhibiting mycobacterial growth and disease if some of those new drugs hit the market. Okay, so nothing, you can't really walk away saying, yeah, these are going to actually take care of the disease. It's just these are the sort of observations that lead people to wonder, and someone's going to have to do the hard work to see if these approaches are going to work out. Okay, so what about RD1 and other non-tuberculous mycobacteria? Starting at the top, NTBH37RV is one of the classic virulent Mycobacteria TB strains that's been in study for a long time. That's clearly RD1 positive. M. kansasii subtypes are typically RD1 positive as well. M. solagi, did I pronounce that right?
I think it's
Zolgai is RD1 positive. Marinum is RD1 positive. M. avium, and this is per our discussion the other day, M. avium has an RD1 but does not have ESAT6 expression. So maybe account for some of the differences in how it functions in vivo. So it's a little bit of an oddball in that regard. Otherwise, this is very scant literature. I think there was all of maybe 2 references where they really compared different strains for the RD1. And in most of these, all they did is confirm they had RD1. They did not confirm that the genes in RD1 were actually functional. So I think there's a lot to be learned about that. Okay, shifting gears wildly. this has been a breaking story since about 2006. The idea that your intestinal bacterial flora, in fact, has a unexpected effect on body habitus. Originally this was in Nature. If it's in Nature, it's got to be right, correct? And more recently, we've learned more about this. So these were originally mouse studies, and what the observation was is you have two major classes. You have several major classes in the gut, but the two we're interested in is one is the Bacteroidetes and Firmicutes. The ratio of these two determines body weight. It associates with body weight, and other experiments have shown that they may actually be causal. Lean mice have an excess of Bacteroidetes compared to Firmicutes, and the obese mice had mostly Firmicutes in a greater proportion than Bacteroidetes. Gut floral transplantation into germ-free mice reproduced the lean, obese polarities based on the ratio of those bacteria. Okay, so it's not simply the obesity somehow generated the change in bacterial flora. This suggests the bacterial flora itself determines the body habitus. And it was wholly unprecedented. So just if you want to try and keep track of this, have a visualization of which bacteria classes cause this. Did this particular person, I don't know who it is, just look at her and think Bacteroidetes. Okay, she is poster child for Bacteroidetes. Oh, well, we can't say that in here. That's. Can we say her boyfriend? She'll want to cut of the profit from ID podcast. Okay, but the sort of bacteria you have, it's your usual. There's no predicting how these. But anyway, so you got Bacteroides, you got Porphyromonas, Prevotella, and a lot of gram-negatives fall in that category. Now, Firmicutes, on the other hand, is the one that makes you fat. And these bacteria include Lactobacillus, Bacillus, Clostridia, and Mycoplasma and a variety of gram-positives. Now, what I find a little amazing is, with all the diversity in each of these classes, there has to be some sort of common mechanism so that the ratio actually has some effect. You'd think that there'd be so much variety that it would all kind of average out, but it doesn't. So apparently there's something class-specific, despite the incredible variety of organisms. And I'm a little blown away about how does that all work. Okay, so this is all mouse studies. So moving on to human studies, and yes, these things, it is moving into human studies. So in 2007, also in Nature. Nature really likes this story, by the way. They have like all sorts of little abstracts and everything they like to talk about. So in human studies, so what they did is they've looked at obese and lean humans, and you see that same relationship. Again, Bacteroidetes versus Firmicutes. Okay, I just don't have a good slide to show that. So what this group did is they identified obese people and put them on 2 weight loss diets, because in this case they didn't want to transfer stool. I mean, no one's done that in humans yet. What they wanted to show is that by changing weight, you would change the ratio of the two. So they put these obese individuals on a fat-restricted and carbohydrate-restricted diet, thinking, well, that's curious. Maybe you restrict one, gives you a different result than the other. Well, down in the bottom right-hand corner there, these patients actually lost weight, a little bit better, I think, with the fat-restricted one, and at the same time, though, okay, so the axis on the x-axis shows you change in body weight, so they all lost weight, and the y-axis is change in the Bacteroides abundance. So since that is associated with thinness, I think I got that straight. Yeah, Bacteroidetes is lean. As they became lean from their diets, they increased the number of Bacteroidetes in their gut. So this seems to hold true, as predicted by the mouse model. It's not dramatic, but apparently it was very reproducible. The very strange thing about this is on the left-hand side, and I'm not going to go into great detail on that, but in those two groups, this is a 16-RS RNA analysis, which basically gives you sort of an idea of the diversity of the flora that those people had. And they didn't really show the data. At some point in this paragraph that was below this, they said, of interest, those patients had, it was almost like a thumbprint, for the sort of organisms they had, based on kind of the genetic variety, and that did not change as they dieted, despite the fact that the two major classes shifted around. So take it for what you will. It's a lot more complex, a lot of bacteria, but still, the observation is that by changing your weight, you change the ratio of the Firmicutes and Bacteroidetes organisms. Okay. So no one really knows quite how this happens. Okay, we're past the point where we have these kind of empiric observations, so we can do some very simple experiments with it. There are a lot of synergistic interactions between our gut and the flora. Obviously, there's a lot of fermentation of unused energy resources that might have something to do with this. Perhaps the Bacteroidetes eats up a lot of the energy that's in the gut that we otherwise don't absorb. It's known that the bacteria in the gut train the immune system in a lot of different ways. It prevents growth of pathogenic bacteria. It regulates the gut development. The gut, it's a very dynamic response. The gut is sensing what's going on in its lumen, and the nature of the bacteria there sometimes can change how the gut develops. Obviously, they produce vitamins, vitamin K, biotin, and they produce some hormones that promotes fat storage. And unfortunately, I couldn't find, this was just a little blurb, and I couldn't find out what that really was. I don't know if it's related to leptin or what. Okay, so we're going to add another little piece to this right now, and that is part of our sensory mechanism within the gut epithelium is constantly looking at the bacteria, and the way that they sense the bacteria are there are through the use of these toll receptors, which I think everyone knows about now. Toll receptor 5 is highly invested in the lumen of the epithelium, the colon and intestinal epithelium, and it has a receptor that recognizes the flagellin monomer. And essentially all the bacteria with flagella have these flagellin monomers. They're reasonably conserved, and toll receptor 5 can identify them, bind to it, and that sends a signal into the cell to do something. And in general for toll receptors, it's a pro-inflammatory signal. And it's, okay, so our gut is constantly in some low-grade inflammatory state. So these investigators said, well, what the heck, toll receptor 5, what's it doing in the gut? So they made mice deficient in toll receptor 5. And toll receptor 5 is mostly involved in the gut itself. You have it in macrophages as well, but that's not the bulk of where the molecule is. So they took these toll receptor 5 deficient mice, and what they found is when you let them go for a while and fed them, the normal rodent chow, they actually became 20% overweight, and were actually markedly hyperphagic relative to wild type mice, which is bizarre because you're just changing this one receptor in the gut, and all of a sudden they're changing their eating habits. They acquired visceral and abdominal fat. They increased their serum triglycerides. They increased, there was evidence of adipose inflammation, which goes along with metabolic syndrome. They had hyperglycemia, increased leptin and insulin, and hepatic steatosis. So these were little poster mice for metabolic syndrome. When they fasted them, and basically just cut the rodent chow way the heck back, they reversed the metabolic derangements, although there was still significant adipose inflammation. So now you've taken this from a model of simply bacteria determining leanness or obesity to now recognizing that there's a third dimension here where the gut epithelium itself is somehow interacting in a way that promotes the metabolic syndrome, which did not occur in the obese mice that had toll receptor 5. All right, so it's a crazy story. It's hard to take home a message from this right now, other than to say this is one heck of an interesting model. And if you think of all the metabolic syndrome people we see in the hospital every day, we can't help but to hope that we're going to find some insights from this that'll be useful. Okay, so the implications obviously are if we can change the gut flora, do we have the opportunity actually to target diabetes and metabolic syndrome? I mean, that's possible. Can we identify genetic differences in the toll receptor 5 itself? Like any expressed protein, you look hard enough, you're going to find all sorts of polymorphisms in either the promoter or in one of the exons, such that it functions differently. And is it possible there's a subset of people who have slightly different toll receptor 5, and that then accounts for the preponderance of metabolic syndrome? What do antibiotics do in this situation? I'm surprised no one's looked at that yet, but I imagine you could probably shift some of the flora around with antibiotics. Is it possible to do bowel flora transplantation as a therapeutic procedure? You're all for it, aren't you?
We might have to.
Okay, and that leads to some very interesting commercial possibilities. So, you know, you don't have to grow these bacteria. You just need to find someone who's thin and get their bowel bacteria.
But she's not always thin.
I think I would choose Taylor Swift over Britney.
Yeah, that's true. Somebody who
Okay.
is innately skinny all the time.
But since it belongs to them, if they're smart, they would have lawyers, and it would be branded. So this would be Brittany branded Bacteroidetes supplement. And operators are standing by. Okay, I should have warned you this was going to be a short one, but